In today's video, we'll be looking at DNA profiling. But before we actually go into the details of it, we need to know a bit more background information about our actual genome. The term genome refers to all of the genes possessed by an individual or an organism, basically the whole base sequence, and that includes the DNA in the nucleus and also in the mitochondria. Amazingly, however, only 0.1% of our genome is different. 99.9% of our genome is exactly the same across all human beings. The remaining 0.1% actually causes all of these variations between different individuals, and we use that in DNA profiling. There are two types of genes within the genome. One is called exons, and the other is called introns. And exons takes about 2% only of the entire genome, and that actually is the bit that codes for our protein, which is a surprising thing considering that our entire body, all the functioning, all the structures are made of proteins. The remaining 98%, which are the introns, in the past we used to call them junk DNA. However, nowadays we don't use that term anymore because we say that they might be involved in gene expression regulation, and that is something that we've actually talked about in chapter 19. Now, when it comes to DNA profiling, we are looking for differences in individual genome. And yes, of course, there will be some slight differences within exons because we do have, different eye colors or skin color, which is caused by proteins. However, for DNA profiling, we'll be looking at the differences within introns. Introns have something called satellite DNA. They are short, repeated DNA sequences. There are two types. One is called mini-satellite and the other is called microsatellites. And the difference between them is that there's another name called variable number tandem repeats, VNTRs. They're a sequence of DNA bases that are about 20 to 15 base pairs long, but repeated 50 to 100 times. Whereas in microsatellites, which we can also call them short tandem repeats, STRs, they are way smaller. They are about 2 to 4 bases long. They get repeated for about 5 to 15 times. So these are bigger repeats than these. But they work in the same way. They are really useful when it comes to DNA profiling because they always appear in the same area on chromosomes. So let's say I have two strands of DNA from two different people. Because we say satellite DNA appears on the same places on the chromosome, so given that this is the same chromosome but from different people, the satellite DNA will also be found at the same place. However, this person has a much shorter satellite DNA, so there are less repeats. Even though the sequence will be the same, but this person has maybe 5 repeats, whereas this person has 10 repeats instead. So if we cut the DNA up when it comes to DNA profiling, you will generate a different satellite pattern. So one of their bands will be exactly the same because of the same length. But let's say if we cut it there, again, this bit will be of the same length, but the remaining bits will be different. This one here will be much shorter than this fragment here. So the satellite pattern will look different. So this is the principle of DNA profiling. Different people have different number of repeats, and therefore they will generate a different satellite pattern, and that is the essential principles of DNA profiling. So for people who are identical twins, then they would share the identical satellite patterns because their genomes are the same. And if people who are closely related would have a very similar pattern, and that's what DNA profiling is. DNA profiling helps identify an individual and determine familial relationships. So a classic use will be in forensics. So let's say we've got different suspects and we've got DNA samples from them, and then we just need to do DNA profiling to compare let's say a DNA sample we collected from the crime scene and the DNA samples from the different suspects, and then we find a match in between. And what we're trying to match is the satellite patterns. Or for paternal testing as well, because the right father would have a very similar satellite pattern or DNA profile as the baby. So that's what it is. So now we'll have a look at the process of DNA profiling. So let's say we're in the crime scene and we have found a pool of blood. We say that the pool of blood is left behind by the killer. So what we want to do is we want to get some DNA from the pool of blood and then compare it to a list of suspects that we have, and so we can find out who the killer really is. So what we need to do first of all is to extract some DNA. The thing is, even from a pool of blood, we can't actually get a lot of DNA from it. What we will then do is to put it through PCR, which is polymerase chain reaction. And that process is basically artificial DNA replication. So in that sense, we will get lots and lots of copies of the same DNA. We'll go for many cycles, make lots and lots of copies of it. So imagine we get hundreds, thousands, or millions of copies. What we then do is to add some enzymes into it. And this particular enzyme is called restriction endonucleases. And we can have more than one, which can cut at different places. And what restriction endonucleases do is that they recognize the specific DNA bases or sequences, and we call them restriction sites. And by recognizing them, so let's say they all have the restriction sites there, then it will then cut the DNA up like that. And another restriction endonuclease will recognize this particular site and then cuts it up again like that. And imagine that lots of them do the same thing. And the point of that is to cut up the DNA into smaller fragments. Restriction sites are actually within the introns, but not on the satellite's DNA. So what happens is it will cut at the intron, cut at the restriction sites, but leaving the satellites intact. That it becomes like this. So if let's say that top bit becomes that bit, the middle bit becomes that bit, and the bottom bit will be that bit there. So what we now get is a little container containing all these different DNA fragments. But we need to find a way to separate these fragments so we can see them individually. So what we then do is to put them onto a gel, and we do gel electrophoresis. So as the name implies, gel electrophoresis meaning we actually put these DNA fragments into a jelly block, and it's made up of agar. And the name electrophoresis is saying that we're going to use electricity. On the agar gel, we are going to have wells on it. And we put these DNA fragments into these wells. So we can do this for the actual sample. So this is our evidence. They're from the crime scene. And we've got suspects A and B. So this process, we'll see if A or B would have the same satellite pattern or DNA profile as the evidence we've gathered from the crime scene. What we'll do is to put these DNA fragments into the wells of the gel here. We'll connect the electrodes together and we'll put it on the negative side of the electrode. And the reason for that is because DNA itself is negatively charged, so it will be repelled from the negative electrode and will be attracted towards the positive electrode. So the DNA fragments are going to move in this particular direction. Another thing to note is we immerse the agar gel in alkaline. So double-stranded DNA fragments here will be broken down into single-stranded DNA. Being single-stranded, they can actually move through the gel a lot easier. So let's say in this particular case, we've got a band that looks like this. And as you can see, suspect A's DNA profile matches with the evidence we've got from the crime scene. I'm drawing it out like this right now, but in reality, when we were doing the actual electrophoresis, we may not necessarily see it because we haven't put in any dye at all. We need somewhere to actually see it. What we then do is we do something called self-blotting. What that is we basically put a nylon membrane or nylon sheet over the entire gel. So imagine if we've got a tray of the electrophoresis tray like this, and then we put the nylon sheet on top of it. What's going to happen is that all these DNA fragments that were separated out is going to get soaked up into the nylon sheet like that. Imagine actually trying to process the data on a agar jelly block is actually quite difficult because you might be worried about breaking it and, alkalines everywhere. So it will be a mess. That's why we do self-blotting, because we can soak up the data onto a piece of paper and we can do further study on it easier. So for hybridization, what we actually do is to add dye to it. So it could be a radioactive dye or fluorescent dye. And let's say usually nowadays we use fluorescent more because it's safer than radioactivity. Then we put it under UV light or X-ray light if we're using radioactive dyes. And then we can see the actual evidence coming out like that. And finally, we can see the evidence presented in front of us. So for a quick recap, we've got our extracting the DNA from the pool of blood from the crime scene, and then we let it undergo PCR to amplify the sample, so we make more of it. Then we add restriction endonucleases, which are enzymes that can cut up the DNA at specific places called restriction sites, and we cut them up into smaller fragments for further processing. Different people would have different number of repeats of satellites, so therefore each person would generate a unique DNA profile or satellite pattern. And we put all the DNA samples into a gel and we let it do gel electrophoresis, where we use electricity or electric current to separate DNA fragments based on their charges and sizes across the gel. We also use alkaline to break down the double-stranded DNA into single-stranded DNA for easier processing, and also allowing, when during hybridization, to allow the particular fluorescent dye or probes to actually attach to the DNA for us to see it. We can then do sulfate blotting, where we transfer the DNA samples onto a piece of nylon sheet or nylon membrane for easier reading and processing of data. Then we expose it under UV light if we're using fluorescent dye, and X-ray if we're using radioactive dyes, and we can then finally see the patterns shown there. And this is DNA profiling.